Nothing. No doubts, people? Okay, I have got this notification that your JEE, NEET and JEE classes, specifically for chemistry and physics, they are clashing with your other class timings, right? With this biology class timing or other class timing. Is it correct? You are getting some extra classes for chemistry and physics. Am I correct, Atif, Fatima, and Hiba?
No, sir, I'm not.
Sorry, what? Can you?
No, sir, I'm not attending classes for NEET and JEE right now.
Okay, you're not attending it for JEE? Fatima and Hiba, are you attending for, sorry, for NEET, not for JEE, for NEET, like the chemistry?
No, sir. No, sir.
So these are the classes whose timing are actually clashing with some of your regular classes. So for that, I was to notify you that if you want to attend these classes, what if these classes will happen at 8:30 p.m. your time, KSA, 8:30 p.m. KSA. So you're not attending it. That's a different thing. Anyone, Hiba, are you attending these? Hiba, am I audible to you?
No, sir, I'm not attending.
You are also not attending. Okay. So if anyone wants to attend of you three, we are planning to have it at 8:30 p.m. KSA your time. This is what I wanted to convey. Okay. Okay, so let's start with today's class. Before we start with the chromosomal theory of inheritance, linkage, recombination, and which is the exception for the third law, let's have a quick recap. So Fatima, are you there? Fatima, Munir, would you like to give a quick recap of what we did in the last class? Where did we start? Law of segregation and law of independent assortment. Okay, quickly, these two things, give me a quick recap. Fatima, are you there?
Yes, sir. Sir, I missed the full last week, so I don't know what's going on because they changed my batch.
Okay, why is that? Yeah, okay. So you were joining the MEA, right? MED, right? Okay. You were taking classes on weekends, correct?
Yes, Saturday,
Yes, and you have
Sunday.
Saturday, Sunday. Yeah, you joined. So will you be like joining this batch now? Because this batch is one class before, like the weekend batch is one
I
class ahead.
want to go back to the weekend batch, but then that batch is clashing with my other classes, like the timing.
Oh,
So
okay.
I
And
don't know.
which other classes they're clashing with, Fatima?
Chemistry. I have chemistry on Sunday and Saturday.
Okay. Okay. So no problem. Like this batch is also like uh, at the same level, like the same chapter. It's just one class trailing. So weekend batch is one class ahead. So probably what you missed in the weekend. Uh, did you attend any class for genetics?
No, sir. The last time
Yeah.
I joined the class, we were doing human. We like finished with the human reproduction.
And
chapter.
what about the chapter 4, the reproductive
No,
health?
I have not. I have not been.
You've not attended that also?
No.
Okay. Okay. So Fatima, let's do one thing. Tell me, do you have any class on Tuesday and Thursday?
Yes, I have physics.
Physics. And what is the timing for that?
4 o'clock starts at 4.
Your time, 4 o'clock, right?
Yes.
Okay, which means 6. Yeah. So can you do 2 classes on Tuesday and Thursday? That way, like you can join a batch which is still like finishing where you left last time. So that batch we have to start chapter 4. Chapter 3 we have finished. That I think this is the last thing you attended in weekend batch as well. So if you can do, after your physics class on Tuesday, Thursday, if you can do a biology class as well, will it be possible for you? 2 hours. That way you can get chapter 4 and beginning of like everything with genetics again. Or at least a few classes you can do there till you come to genetics, like for chapter 4, and then you can continue this batch. What do you think?
Yeah, I can do that.
That will work, right? A few classes to just give you a backup,
Yes.
right? So it will be your backup and the regular batch for them. Okay, Fatima?
Yes.
Okay, so that will work. Very good, cool. Okay, so Atif, I think I'm left with Atif and Hiba. Atif, would you like to give me a quick recap from law of segregation onwards, just quickly?
Yes, sir. 1 minute.
Yes. So Fatima, we will be doing chapter 5, genetics today, where we have studied the laws of Mendel, the 3 laws: law of dominance, law of segregation, and law of independent assortment. Okay. So Atif will give you a quick recap of law of segregation and independent assortment. And today we will start with a new concept called chromosomal theory of inheritance and linkage, so that you can connect to. Okay. Atif, whenever you are ready. Okay. And all three of you have been added to the group. There is a group, WhatsApp group with me. Fatima, Hiba, Atif. Are they in the group, right?
Yes, sir. Sir, may I start?
Yes, Atif, please.
Okay, sir. So in the law of the law of segregation, alleles do not blend during the gametogenesis, and each gamete will receive one allele which is segregated randomly and independently of each other. Then the law of segregation is the only law that is universally accepted with no exceptions.
Yeah.
Then
Yes. And would you like to explain this part? What did we discuss here with P, Snapdragon, and the blood?
yes, so the PP like follows the normal dominance, that is the Mendelian inheritance. Like there is one dominant allele and one recessive allele, and only the dominant allele is expressed if it is inherited. Then the Snapdragon follows incomplete dominance, where like both the alleles are not able to express themselves completely, and it ends up being like a mix of both the uh, traits. Like for example, capital R is red and small r is white, but capital R small r is pink. And then blood follows codominance, that is
Yeah.
two alleles control like a single trait, I think. So in
So
blood there
both,
is
both the alleles are equally expressed. Okay.
yeah, both alleles are equally expressed. Yes.
So this part,
So
I was just telling you that whatever be the condition, whether there is dominancy, there is incomplete dominancy, or there is a codominancy, but the law of segregation holds true all the time. That's why it is called law of purity of gametes. These alleles always segregate independent of each other whenever they have to make gametes, right?
yes, sir.
Yeah. Okay. Continue. Next. Then we started with 2-gene inheritance, right? What is 2-gene inheritance? Atif?
Two-gene inheritance is like the study of like the inheritance of two characters. Like, yeah, for example, instead of studying just one set of alleles like capital E, we will study two set of alleles. So yeah, we will cross
Yes.
both of these using the Punnett square.
Yeah.
And
Right.
when we cross them with the F1 generation, all were so we crossed a pure pure violet flower and a dwarf white flower. And the F1 generation, all of them are hybrid tall and hybrid violet. But when we cross the self-cross the F1 generation, we got the phenotypic ratio is to be 9:3:3:1, and the genotype ratio to be 1:2:1:2:4:2:1:2:1.
Yes. Okay. Thank you, Atif. So I'll take from here. So this
Yes,
is
sir.
to explain. Yeah, thank you so much. So to explain a little bit more to Fatima and Hiba, you're there. Can you hear me? Yes, to both of you. See, basically, two gene inheritance is, as Atif correctly said, instead of one character now, we are talking about two characters being inherited together. And this is what normally happens in biology. It's not like in biology, every one gene is inherited alone. All the genes are inherited together, when gametes are formed. So basically, when two genes are inherited together, they may also have a connection or a relationship between them. And to understand that, the interaction between two genes, we have to study two gene inheritance, which we can study through dihybrid crosses. Now in dihybrid cross, we, as it says, we take two characters. One is this pure tall and violet, and then dwarf and white, right? So these are two genes. For pure tall, capital T, for pure violet, capital V. And similarly, and we make a cross to figure out what happens in F1. So in F1, all will be hybrid tall and hybrid violet. Okay. This is following the law of dominance like it was following in single gene inheritance. But interesting thing happens after this. So when you self cross F1, you find that now instead of 4, like there are 16 combinations that come up, because each of the parent in the self cross can make 4 different gametes combination. These are the 4 different gamete combinations. On the other hand, also there are the same 4 different gamete combinations. And when they fuse together, they form 16 progeny combinations. And there the genotypic ratio, remember what was the genotypic ratio in a monohybrid cross in the F2? 1:2:1, remember? In the monohybrid cross, where we were studying only one gene, it was 1:2:1, correct? Total 4 offsprings were there, 4 combinations were there. And what was the phenotypic ratio? 3:1, right? 3 were tall, out of which 1 was pure tall, 2 were hybrid tall, but 3 were tall and 1 was dwarf. Are you remembering that, Atif, Fatima, and Hiba? I hope I'm also audible clearly. If not, please let me know. Atif?
Yes, sir.
Yeah, okay.
Yes, sir.
But here the genotypic ratio is
Sir, did
of
you call
6.
me?
Yes, I was saying that, am I audible? Like, do you understand what I'm saying?
Yes, sir, audible, sir.
Yeah. So the genotypic ratio here in a dihybrid cross is given for 16 combinations which are formed. So it is 1:2:1:2:4:2:1:2:1. So these are the genotypes. If you start going from diagonal, from one diagonal we start, this is just one unique genotype, then 2, then again 1, then 2, then these 4 are same, then 2, 1, 2, and 1, right? So it starts like 1, 2, 1, 2, 4, 2, 1, 2, 1, total 16. And the phenotypic ratio is 9:3:3:1. Now in phenotypic ratio, basically what are these phenotypes? This is tall and violet. Okay. This is tall and white. This is short and violet, and this is short and white. Okay. You understand? Everyone? Then we also studied law of independent assortment, which is the third law given by Mendel. And this law states that the alleles of 2 or more different genes, when now because we are studying 2 gene interaction, so when 2 or more genes together are inherited, then the alleles of these different genes get sorted into gametes independently of each other. Okay, which means no gene affect the inheritance of other gene. the inheritance of both the genes are independent of each other. This is what this law says, but this is not a universal law, and this law has exception, and the exception is linkage between the genes. Now, this is saying that if I take two genes together, one gene will go separately of the other gene all the time. But sometimes this does not happen. Sometimes some genes have linkage in a way, like you can understand this as two best friends. So wherever one friend goes, the other follows, right? Or they both go everywhere together. So if you spot one of them in a party, then the chances of other being in the party is also very high. Am I correct? So that is called linkage between genes, which Mendel did not know when he did the experiments on P. Now, to understand what basically is, before we went to study linkage, I gave you an example, a question where we crossed a heterozygous round and homozygous tall with a wrinkled and a short plant. And we realized that depending on what are the genotypes of the parents in the next generation, both the genotype and the phenotype will be different, right? So these were some questions that we did. And then finally, we talked about some board questions that come in board. One of the question was that why did Mendel's work remained unrecognized for very long? And it was because of three reasons. One, communication was not easy, so work could not be widely publicized. And Mendel's concept of gene was not accepted by the contemporary scientists of that time. And third, he used mathematics to explain biology, which was a very new thing to do and was very unpopular. And scientists did not accept it. In the end, I told you that there were three scientists who rediscovered Mendel's work independently: Hugo de Vries, von Schermack, and Correns. And these three scientists raised the work to the status of laws. So these laws that we studied were made laws by these, especially Hugo de Vries. Okay. And hence today we will start with chromosomal theory of inheritance. And let's begin. Okay, so you all can see the screen. So let's start with chromosomal theory of inheritance. Write down. What do you understand by chromosomal theory of inheritance? So Mendel, when he did his when he did the research work, he actually did not know about genes. Okay, so that there are genes in DNA, so he called them factors. Correct? Yes, Atif?
Yes, sir.
Eba? Yeah. So these, when he did not know about genes, clearly he was also not aware that these genes are actually a physical, like they are present, they are a part of DNA, they are a physical region of DNA itself, and they are present in the form of chromosomes, which are inherited as discrete units. So chromosomal theory of inheritance, write down, it was given by, chromosomal theory of inheritance was given by Sutton and Boveri. So there were two scientists, Sutton and Boveri. They gave this chromosomal theory of inheritance. And this says, write down, this theory argues that the pairing and separation of a pair of chromosomes, pairing and separation So whether it is pairing or whether it is separation of a pair of chromosomes would lead to the segregation of a pair of factors, in bracket write down genes, a pair of factors that they carry. So you understood what basically they are trying to say? They are trying to say that what Mendel called factors are basically genes which are present on chromosomes. Okay, so if we talk about inheritance, it is the chromosomes that are being inherited. So this is the physical thing which we can see with our eyes, that is getting from one parent to another, and which is carrying all the characters and traits. Is it clear? Fatima, Atif, and Hiba?
So it is just an updated version of Mendel's law of segregation.
Exactly, yes, because Mendel in his work, in his theory, did not talk about anything. He could not explain what are these factors. So he said, he wrote capital T, small t, but he did not know what is that capital T, what is allele, what is gene. Now, when we know all of this, it becomes easier for us to understand Mendel's work. Imagine if I were teaching you Mendel's work the way he wrote, you also would not be very convinced and would not like to study it. And that is what was the issue with the biologists of that time, because Mendel did some pure mathematics, some concepts of ratios, crosses, and he said that certain factors come from one parent to another and they do not blend. When he said they do not blend, then it became the law of dominance, but it had exceptions, and we know that they blend in incomplete dominance and they sometimes express together in codominance. Then one thing he said was that these factors, when during gamete formation, always segregate independently, and that what he said was correct. It does not have any exception. So that was law of segregation. Then the third thing he said was that these factors, when we study multiple genes or multiple characters, then these factors segregate independent of each character. Like the factor for tallness will not affect the factor for color of the flower, for that matter. That is law of independent assortment. So this work was updated to the level of to the status of laws later on. Similarly, when he was talking about all these factors, he did not know what are these factors. So we know that those were genes. And when Sutton and Boveri came, they said that whatever Mendel did, it is correct. And basically, it is the chromosomes that are being segregated. So people asked, how can you say they are chromosomes? Then they gave this reasoning, which this theory also states, that if you separate chromosomes, then you will see that you are also separating the factors. You understand, Atif? If you pair chromosomes, you will see characters are pairing up, which means one character was present in one chromosome, the other allele was present in another chromosome, one from father, one from mother. When you pair them, you will see pairing, a heterozygous condition. When you separate them, you will see gamete formation or a single allele. Do you understand?
Yes.
Yes. So yes, this is an updated version and a more concrete version of Mendel's law of inheritance, which is known as chromosomal theory of inheritance. So write down Sutton, this theory, Sutton through this theory united the knowledge of chromosome segregation, chromosomal segregation, because by this time we knew that chromosomes segregate. So this theory united the knowledge of chromosomal segregation with Mendelian principles of inheritance. With Mendelian principles of inheritance to give a combined chromosomal theory of inheritance. To give a combined chromosomal theory of inheritance. Is it clear? Atif, understood?
Yes.
Okay. So Sutton and Boveri gave the theory, but still it was not proved. It needed to be proved through some experiments that can actually tell us that yes, when chromosomes segregate, factors segregate. When chromosomes don't segregate, factors also do not segregate. So write down the experimental verification. So you have to remember these two scientists for giving this theory. You have to remember Mendel for doing all the work. You have to remember the three scientists, Hugo de Vries, Karl Correns, and von Schermach, for raising the work of Mendel to the status of laws, especially Hugo de Vries. And then you have to remember this one, another scientist. Keep making a star in front of the name of the scientists and their contributions, because that is asked in entrance and boards both. So there was another scientist called Thomas Hunt Morgan. Thomas Hunt Morgan. He experimentally verified the chromosomal theory of inheritance. He experimentally verified the theory, the chromosomal theory of inheritance. Okay. Is it clear? Now, while Thomas Hunt Morgan was doing the experiments, he did the experiments, write down. He did genetic experiments on Drosophila. This is the name of the organism. Drosophila. It is commonly known as fruit flies. If you do not remember Drosophila, it's fine. If you remember, very good. But remember fruit flies. These are tiny, tiny flies which actually hover around the fruit peel and anything that smells sweet, like banana or any other fruit, even especially mango and citrus fruits as well. So you can see these small flies. Sometimes people get confused. People confuse them with babies of housefly because they are very small. They are not babies. They are adult. They are grown up, but they are tiny as compared to the housefly. Okay. So Thomas Morgan did experiment on Drosophila, the similar kind of experiments that was done by Mendel on pea plant. So basically Mendel also looked at characters in the pea plant and figured out how are they inheriting. Yes, right, from one generation to another. Yes or no?
Yes.
Yes. And luckily Mendel could not find any incomplete dominance, any codominance or any linkage there. But when Morgan did the similar kind of experiments, he figured out that there is linkage between some genes. Some genes do not inherit following the laws of Mendelian inheritance. Now write down, let's talk about linkage. What is linkage? Write down linkage of genes. Now what does the word tells you? Linkage. Does it tell you something? What does linkage means?
Like being together.
Yes, genes are linked together somehow. Now let's figure out how. How can we first experimentally prove that they are linked? So for that, write down Morgan crossed yellow-bodied and white-eyed female fruit flies, yellow-bodied and white-eyed female fruit flies with brown-bodied and red-eyed male fruit flies. Now, it's not like he already knew that he will do this and he will find something interesting. He was just trying to do the similar kind of experiments that Mendel did on P, right? Mendel did tall with violet flowers. Remember the dihybrid crosses we did just above? Atif? This is also a dihybrid cross, right?
Yes.
Having two characters, but it is in Drosophila, it is in flies. So when he did that, he thought that it will be the same thing. In first generation, you will find all the dominant traits, and in the second generation, when you will cross them, you will find the ratio of genotype 1:2:1:2:4:2:1:2:1, and phenotype will be 9:3:3:1, correct? But this is not what happened, and he thought that maybe there is some he committed some mistakes while making the crosses, or while putting, there is some contamination or something. So he did it again, and again it did not follow Mendel's ratio. After multiple times, he realized, okay, if they're not following Mendel's ratio and there is no flaw in my experimental design, then maybe Mendel's law of independent assortment is not a universal law. It has a exception, and this was the exception where these two genes, the yellow-bodied gene was actually linked with the white-eyed gene. Okay, so the gene for the body color was linked with the gene for the eye color. Okay, and these two genes always used to stay together. So if a fly is yellow-bodied, most of the time that fly will be white-eyed also. So write down, when you cross this, and then intercrossed or self-crossed their F1 progeny. Intercrossed and self-crossed their F1 progenies. He figured out that the F2 phenotypic ratio, the F2 phenotypic ratio deviated from Mendel's ratio. Is it clear? So Mendel's ratio was 9:3:3:1, correct?
Yes.
Now in if you see in yellow-bodied and white-eyed and brown-bodied and red-eyed, there are dominant and recessive genes, right? So they should have been segregated finally in this form in F2, but it did not happen. This proved that there is a physical association. This proved that there is a physical association or linkage between these two genes. Between these two genes. Is it clear?
Yes, sir.
Now, when the concept of linkage comes, so you can write down, so Morgan only gave the term linkage. Write down, the term linkage was given by Morgan. Because he only for the first time realized that law of independent assortment is not true all the time. Term given by Morgan. Is it clear? Now, if there is a linkage between genes, the next question comes, is there linkage between other genes as well? And if there is linkage, then what will happen? There will be no variations, because see here, following the 9:3:3:1 ratio, it means that 9 and 3 were the parental genotypes. Yes, 9 and 3 here were the parental genotypes. Let's go little up and look what's happening. Normal ratio. Yes, look here. What were the parents which we crossed, self-crossing? This parent was tall and violet. Yes, everyone, pay attention here.
Yes, sir.
Parent was tall and violet. So tall and violet is the parental combination. Am I correct?
Yes, sir.
This combination, of course, will go as it is in the next generation. When we do the F2 cross, you will see that 9 are tall and violet. Then 3 are tall and white. Was tall and white any of the parent tall and white? Even the parent generation was tall and violet or dwarf and white. So what is the parent combination? Tall and violet and dwarf and white. Yes.
Yes.
So parental combination, tall and violet and dwarf and white. But how many are tall and violet and dwarf and white? This 9 is tall and violet. So this is parental combination. I'm writing PC. And this is dwarf and white. This is parental combination. I'm writing PC, right?
Yes.
But was any of the parent anywhere in the cross? Was any of the parent tall and white or dwarf and violet? No. So these two actually are recombinants. You understand? They are new recombinations which are formed when you follow law of independent assortment, right? So these 6 out of 16 are recombinants. Yes or no? That's how this ratio came up. Do you understand now, everyone? Atif, Heba, and Fatima?
Yes.
So similarly, in the case of Morgan's experiment also, it should have been followed. There should have been some parental combination like yellow-bodied, white-eyed, brown-bodied, and red-eyed. But there should also have been yellow-bodied and red-eyed and brown-bodied and white-eyed, right? But those flies were not found, or were just 1 or 2, which means there is something happening which is not allowing recombinants to form, and that was linkage. So when linkage is there, recombination is less. Do you understand the relationship between linkage and recombination? Linkage is inversely proportional to recombination. Is it clear, everyone?
Yes, sir.
So more the linkage between two genes, less will be the new variations or new recombinants formed. Do you understand?
Yes.
Yeah, now let's try to look how is it happening. Okay, so for that, everyone pay attention to this thing here. Let's start with a, so before we understand this, we also have to understand few concepts. This is clear, linkage is clear, how Morgan proved it is clear, because I'm going to tell you some concepts now. If you have any doubt, please ask
Yes.
me. No doubts? No? Okay. So write down about chromosomes, write down homologous chromosomes. You know what are homologous chromosomes? Anyone? So this is a chromosome, okay? This is how it is. Let's say when it is blue and it has a centromere, black thing in the center. Let's say the another chromosome is green. I'm using different colors just to segregate between these two chromosomes. And it also has a centromere. Now in a chromosome, if this is chromosome number 1 from the mother and this is chromosome number 1 from the father, so basically they are same chromosomes, right? When you are pairing up as diploid, one chromosome comes from the mother, one from the father, right?
Yes, sir.
If both the chromosomes are basically same from different parents or sexes, we call these two as homologous chromosome. Is it clear?
Yes, sir.
Yes. Now when chromosomes come, they are like this. So these two things are known as chromatids, and every chromosome has two arms. In the center is centromere. Now it can be exactly in the center or it can be slightly away from the center, up or down. Depending on that, there are two arms. The longer arm is known as Q arm of the chromosome. Okay. Is it clear? And the
Sir, can you repeat
short,
once more?
depending on the position of centromere, so centromere is not always exactly in the center. Okay. And depending on this position, there are two arms of the chromosome, Q arm and P arm. Okay. The shorter one is called P arm and the longer arm is called Q arm. Is it clear? Of the chromosome.
Yes.
Yeah. It is called P because P for petite. You know what does petite means? Petite means tiny, small. So petite arm is P arm and Q is the longer arm. Same is here. So this is the Q arm and this is the P arm. Is it clear?
Yes.
And this is how we note it. And on these arms, there are genes which are located. So for example, say there is one gene here. So the same gene will be present here. Can you see this yellow mark which I'm making?
Yes, sir.
And if there is another gene here, then the same gene will be present on the other chromosome. Can you see this red mark also?
Yes.
Yeah. And if let's say some other color I'm using, if there is another gene here, it will be same as on the other homologous chromosome. So basically homologous chromosome are identical. They have same number of genes at the same location on the same position with same length of Q arm and same length of P arm and the same positioning of the centromere. And also the total length of the chromosome will also be same. Is it clear between homologous chromosomes?
Yes.
So write down homologous chromosome are identical chromosomes. Homologous chromosomes are identical chromosomes with same length, centromere position, same length, centromere position, same staining pattern. We can stain the chromosome. When you put any color on chromosome, it makes a pattern, a band, and that pattern will be same staining pattern, same genes, same number and location of genes. Okay, so basically homologous chromosomes are same. That's why they are homologous. If they are heterologous, they are different. Chromosome 1 from one set and chromosome 2 from another set are heterologous chromosomes. Is it clear? Atif, Heba, and
Yes,
Fatima?
sir.
Yes. Now, these chromosomes, when they are like inherited, basically they have one chromatid. So this one stick is called a chromatid. And these two here, because they belong to two different chromosomes, so they are known as non-sister chromatids. Is it clear?
Yes.
So what is a sister chromatid? Just before division, just before cell division, remember earlier in the classes I told you that whenever cell division used to happen, even in mitosis and meiosis, chromosome number doubles and then divides, right? Especially in
Yes, sir.
meiosis 2. Meiosis 2 is just like mitosis, and in mitosis also. So how it doubles is like it forms one copy of itself, of both the chromatids. It forms a copy. So it from one, it comes into this X-like structure. This is what we think as chromosomes in the books. Books make these, but what they don't tell is basically it's the replicated part, right? You understand, everyone?
Yes, sir.
So just before division, there are two chromatids in every chromosome. And these two chromatids are known as sister chromatids. Okay, because they are from the same strand. They are just copies of each other, exact copies. You understand? So chromatids of the same chromosome are sister chromatids, and chromatids of different chromosomes are non-sister chromatids. Is it clear? You have to remember this very importantly, because now I will be telling you how linkage happens and how recombination happens. Please draw this. Okay, and this thing is, let's say, gene 1, 2, and 3 with colors that I have shown. It is gene 1, 2, and 3. So if we talk about the yellow, let's say this yellow one, it contains one allele for, it contains the gene for tallness, and the mother is containing capital T as a allele, and father is containing small t. It's possible, right? Let's say the second gene is for flower color, and mother is containing red, capital R, and father is containing small r for white. Okay. And this third gene here, let's say for something else, yellow and green, something, another color like pod color or something. So this is capital Y, and this is small y. You understand? On the other strand, which is sister chromatid, which is produced, you can just understand that the same location will be here also for every gene. Yes or no? Because it's just the copy.
Yes, sir.
Yes. Now, if I ask you the question that on sister chromatids, is it possible that two different alleles of the same genes can occur? Say yes or no. Is it possible to have two different alleles of the same gene on sister chromatids?
No.
Atif, is it possible? What do you think? Hiba?
No, sir, I don't think it's possible.
Why you don't think it's possible? Why isn't it possible?
Because when the sister chromatids are formed, the DNA replication takes place. So it should be like,
Exactly. So it is the
yeah.
same DNA, right? It is the same DNA strand carrying the same genes and the same alleles. So on a sister chromatids, it is not possible to have two different alleles of the same gene. But on non-sister chromatids, it is possible. Can you see this? The gene is same, but one is carrying a dominant allele, another is carrying a recessive allele. So different alleles for the same gene, write down, are possible only on non-sister chromatids of a homologous chromosome. Write down this statement. non-sister chromatids of the homologous chromosome. Just 1 second. Who is please draw this, label, mark everything, and let me know. I'll move forward. Done, people. Right. When you are done noting this, please let me know.
Done, sir.
Done, Atif. Fatima and Heba. Fatima and Heba, I'm waiting for you. You're done, Heba?
Done?
Yes,
sir.
Yes, Fatima? Done?
Yes.
Yes. So you understood what are homologous chromosomes? Okay. What are sister chromatids? What are non-sister chromatids? P arm, Q arm, centromere. And this location where genes are present, so this capital Y and small y is present on the same location. This location is actually known as locus. So write down the position of gene on a chromosome is known as locus. Plural is loci. Okay. So this is the first locus, second locus, third locus. You understand, everyone?
Yes.
Yeah. Now let's move forward and let me use the same chromosomes again. So what happens during normally when, how do we make variants? So do you see here? Look here carefully. Now there are 4 chromatids. Can you see 1, 2, 3, and 4 chromatids? Yes. 2 chromatids will have the combination capital T, capital R, and capital Y. These 2 chromatids, the blue one. Yes. And rest
Yes.
2 chromatids will have the combination small t, small r, and small y. And both these combinations are parental combinations. Yes. Because they are coming from the parents in the form of the chromosome. Now, how does variations are formed? Variations are formed due to an event which happens during meiosis known as crossing over event. So write down crossing over of chromosomes. And this actually happens physically where? So I'm using the same chromosomes. Now you can figure out that this is the chromosome, right?
Yes.
Parent one. This is the blue one. Color it blue. This was from the mother, right? And what was the combination this blue chromosome was carrying? Please use colored markers for your notes also. So what were the color combinations it was carrying? It was carrying a yellow gene here, right? Let's say here. Then what was the other one? Green, right? Sorry, red. So it had one gene here. Then it had another gene close by. And there was one more gene, right? Which was towards this end. Now, tell me what will be the alleles? What will be this allele? Capital Y. So this was capital Y. What will be this one? Atif. Capital Y. Because they are sister chromatids, correct?
Yes, sir.
Yeah. If this was capital R, what will be this one? Capital R. And if this was, what was this one? Capital T. So capital T and capital T. Can you see this? Okay.
Yes, sir.
And there was other one which was green. Let's make the green one also because they both are present together. Tell me quickly the alleles carried by the green, the other chromosome.
Small t, smaller, and smaller.
Yes. So it will be small t, small r, and small y, right? So what happens is during crossing over, how recombinants are formed is like this. The one homologous chromosome forms a recombinant crossing over. With the other chromosome. You see here, everyone?
Yes.
So Color it. Color is important because this is going to give you the understanding. So this region here of the blue chromosome, you can see, is showing a crossing over, right, with the green chromosome. Can you see here?
Yes,
And
sir.
crossing over here is happening between sister chromatids or non-sister chromatids? Answer.
Non-sister chromosome.
Yes, between two different chromosomes and non-sister chromatids. This is the centromere for both. And here also, due to this result, let's say capital R, small r, small r. Capital T, small t, small t. And what was the third gene? So the third gene which was present, capital Y, and here
Capital.
it was capital Y, small y, small y. Now due to this recombination event, from where the overlapping is happening, from that point, the two chromosomes physically are cut. Okay, they physically gets cut and rejoined. So as a result, when they get cut and rejoined, you will see this happening. In the blue chromosome, which was from the mother, this thing will be intact. But from here onwards, there will be a green part, right? You understand, everyone?
Yes, sir.
Because it formed a recombinant now. Now, can you see that this chromatid, which I am currently coloring, it is a recombinant chromatid? Whereas this chromatid, these two are sister chromatids only. But now one is parental and one is recombinant. Yes or no?
Yes, sir.
Now, interesting thing. Let's write the alleles. So same capital T, capital T, capital R, capital R. But now there will be capital Y and
Small y.
small y, right? Is it clear, everyone?
Yes.
Now, if we make the other one, that also will be like this. So one chromatid will be okay. But the other chromatid will have a blue region attached. Yes?
Yes, sir.
Let's color it. So this is what exactly physically is happening in chromosomes. When our chromosomes are forming variations, that is why it only happens during meiosis. And did I tell you that meiosis only happens during gamete formation in sexual reproduction? And whatever we studied in reproduction chapter, can you now connect that concept here? When I told you that sexual reproduction creates variation, this is what I meant, right? This will be
Yes.
small r, small r. Genetics is the basis of what's happening during reproduction, small t, sexual reproduction, to give rise to variations. So this is how variations come up. In this case, we here have just realized it for one gene, and this is small y, but this is capital Y, right? Now look here carefully, people. This is chromatid 1, right? This is chromatid 2, this is chromatid 3, and this is chromatid 4. Yes? Everyone understood? Out of which chromatid 1 and chromatid 4, what was the combination? Capital T, capital R, and capital Y, right? Same, small t, small r, and small y, right? These two are parental types. You realize?
Yes, sir.
So 1 and 4 will be parental combination. And finally, there will be segregation to make haploids in for next generation, the gametes. These two will segregate and go into one gamete, right? So they will be parent. This will be the parental combination. Whereas if you look for 2, second one here, I'm using another color for this. What is the combination? Capital T, capital R, but small y. And for this, small t, small r, but capital Y. Do you understand?
So,
Yes. So 2 and 3 will be recombinants. Do you understand now how variations come up in sexual reproduction?
yes.
Yes, everyone? Is it clear, everyone? Fatima? Fatima and Hiba, is it making sense to you?
Yes, sir.
Yes. Please mark it, write it down. So this is crossing over. And this part here, sorry. Yes. This part here where this crossing over is happening is called chiasmata. Chiasmata or crossing over bridge. Crossover bridge or recombination bridge. Is it clear? But basically in books it's known as chiasmata. This part, this region here. Okay.
Yes.
And if you see, first write down this, make and write down this, and let me know when you're done. And I'll tell you one more concept. Let me know when done, people. So these two are sister chromatids. These two are also sister, right? But chiasmata is formed between these two are non-sister. Is it clear?
Yes.
And these are different genes that we are studying. These two chromosomes are homologous chromosomes, correct? You understood it.
Yes, sir.
So at this step, at the chiasmata step, there is exchange of genetic material. Okay. Between the non-sister chromatids. And in this one, parental and recombinants, non-recombinants and recombinants are formed. So parental are also known as non-recombinant chromatids. Recombinants one are called recombinant chromatids. Okay.
Yes.
So look, everyone done? Hiba, Atif, and Fatima? Drawn and written everything. You can take a screenshot if you want. Shall we move forward?
Not-
Yeah. Okay. Good. Okay. So now you already know linkage and recombinance relationship with each other. What is the relationship?
Linkage is
Linkage.
inversely proportional to 1 by recombination.
Yes. Recombination. So more the linkage, less the recombination. Less the linkage, more the recombination. Yes. Now all of you has NCRT with you. Everyone. NCRT right now. Hiba, Fatima, and Atif, do you have NCRT with you right now? NCRT,
Yes,
the
sir.
book. Yes, Hiba has. Fatima. So all three of you have, then we'll do an activity. Fatima and Atif, do you have NCRT with you?
Yes, sir.
Atif, what did you say? Yes or no? Atif, I could not hear your answer. It was very quickly muted.
I have it, sir.
You have it? Okay, cool. So come to page number 84, everyone. Page number 84. Sure, NCRT. Everyone open page number 84.
Yes.
Do you see there are two cross A and cross B, two crosses shown? Do you see this? Keep saying yes or no if you see this. Let's focus on cross A first. Okay. Look at cross A. The female. Okay. So this is about the Drosophila cross and the parental type as yellow and white. So this gene yellow and white, which you see, is present on the chromosome X. And on the other side, you see for males have only one X chromosome. So other one is just shown as an empty bent bar. Can you see this? So do you see this type of arrangement? There is one chromosome like this, the other like this in female. In male, it is this. Can you see this? Right?
Yes.
Now here you have Y and W very close to each other, right? Y and W.
Yes, sir.
In the female. On the male, you have Y plus and W plus, right? Plus is for wild type. Okay. And non plus is for the non-wild type characteristics. So if
Sir,
Y
what
and
exactly
W,
does wild type mean?
wild type means which has no mutation, no change, and which is present in the wild. So this yellow and white have mutated from the wild type as a trait. So if Y and W is for yellow and white, Y plus and W plus will be for brown and red. Okay. That NCERT
Okay,
is
sir.
not written. So write down yellow and white. On the wild type side, write down brown and red. So Y plus is for brown and W plus is for red. We can also use the notation capital Y, small y, but they have used Y and Y plus. Okay. So parents
Okay.
are there. When you make crosses, do you see that one strand has been interchanged? This is not crossing over yet. It is just the exchange of one chromosome. Do you see the female now is one yellow and one blue, which came from the male? Yes. In the
Yes,
F1
sir.
generation, because gametes got formed and they again got together. When you self cross F1, this is where your parental and recombinants come. So look here in cross A, because how many are parental types? What is the percentage? Parental is 98.7%. Can you see, everyone? Are you following? Fatima,
sir.
Hiba? Yes. 98.7% is parental. And how many are recombinant? 1.3%. So what does this tell you about the nature of these genes? If 98.7% of progenies are parental combinations, this means that these two genes are linked together, right?
Yes, sir.
And these are the same genes that Morgan also studied. Yellow-bodied white eye and brown-bodied red eye. And he did not
Yes.
receive Mendel's ratio. This is because, this is the reason. And do you see that Y and W, both these genes are very, very close to each other on the chromosome?
Yes.
Now go to cross B. The first difference that you will see between white eye and miniature wings. So these two are for, write down, white eye and miniature wings. W is for eye color and M is for wing shape. So white eye and miniature. The cross is happening the same way. The recombination is also happening the same way. But do you see there are two differences? One is the distance between the two genes is more, right?
Yes.
Yes. And because of the more distance, how many are parental type now?
62.8%.
And how many are recombinants?
37.2%.
Has the recombinants increased steeply?
Yes.
And the B1, the cross B1 is following 9:3:3:1. Yes or no? Out of 16, if 10 are parental, what is the percentage? Tell me. What is 10 out of 16? What is the percentage? Calculate the percentage. In 9:3:3:1, 9 and 1 are the parental and 3 and 3 are recombinants. So 6 are recombinants, 10 are parental. What is the ratio? What is the percentage of parental combinations in the phenotype? 9:3:3:1. How much?
62.5%.
Yes, 62.5%. And what will be the recombinants then? 37.5%, right?
Yes.
So do you think that cross B is following Mendelian genetics?
Yes, it is very close to following Mendelian.
Yes. So 62.5 and 62.8 is like the same thing, you know. We just consider them as same. They are following the same ratio. When you will make it as a ratio, it will be 9:3:3:1. Then only 62% and 37% will come. You understand? 63% and 37%. So cross B is following Mendelian genetics, whereas cross 1, write down, cross A is not following Mendelian genetics because of linkage between the two genes. So there is no linkage between the genes in cross B, but there is linkage between the genes in cross A. Is this clear, everyone?
Yes.
Okay. No doubt.
No, sir. No.
Hiba, Fatima, please ask me if you have any doubt from linkage onwards. Chromosomal theory of inheritance, linkage. Go through all the notes of today's class once again and ask me if you have any doubt, because now I'm going to switch tracks. We were studying about Mendel till now. We are going to switch tracks and study something which Mendel did not cover. So something non-Mendelian genetics. So this was till now was Mendelian genetics. Please go through your notes. I'll come in 2 minutes. I'll take a quick water break. Is it okay, Atif? I'll be just back. Quick water break, Atif.
Yes, sir.
Yeah.
Yes, sir, it's fine.
Just go through the notes and let me know if you have any doubt.
Okay.
I'll be back. Okay. Hello, everyone. I'm audible and visible.
Yes, sir.
Any doubt in linkage, recombination, chromosomal theory of inheritance, crossing over, homologous chromosome, non-homologous chromosome, sister chromatids, non-sister chromatids, recombinants, non-recombinants, anything? Chiasmata, anything? No doubt?
Sir.
Okay, perfect. Now let's talk about sometimes. So till now, what we have studied that for every character, there is a gene, right? And in every gene, there are more than one alleles. Yes?
Yes.
So this is the Mendelian genetics, and this is the normal course of genetics. Now there is something which is known as polygenic inheritance. Polygenic inheritance. Okay. It is it's not like multiple genes are being inherited that we have studied in dihybrid and trihybrid cross. That is multiple genes being inherited at the same time, but there each gene is responsible for one or the other character. Is it clear? How is polygenic inheritance different? Write down. There are many traits. There are many traits for which two contrasting alleles are not possible. Only two contrasting alleles are not possible. For example, in case of Mendel, when we studied tallness for pea plant, there were two alleles, capital T, small t. So the plant was either tall or short. Yes or no?
Yes, sir.
And even if we consider blending, incomplete dominance, then the plant can either be tall, medium, or short. Again, there are fixed heights, right? Either 2 foot or 4 foot or in between 3 foot, but distinct heights are there. Is it true for human height? Do you think it is the same for human height?
No, sir.
Do we also have distinct heights in which everyone falls? No, right? We have a gradient and every height is possible, right? Every foot and every inch. Yes or no? It's possible.
Yes.
And same is in the case of skin color, right? It's not just humans are black or white or black, white, and brown. We are all shades of the tones. Yes or no?
Yes, sir.
So these kind of characters are actually under polygenic inheritance. There are multiple genes that control this one trait. Okay. And for that, those multiple genes also, there are multiple alleles. Okay. So 2 alleles for each gene, and there are multiple genes. So there are around 5 to 6 genes discovered till now for human skin color. Now, a very complex interplay between these genes gives the gradient. And apart from that, you know why there is a skin color in humans? What is responsible for it? What is responsible for skin color in human?
Melanin.
Melanin. What is melanin?
It's a hormone.
It's a hormone. No, melatonin is a hormone. Melanin is a pigment. Melatonin is secreted. You know, you're getting confused between melatonin and melanin. Write down. Melatonin is a hormone that helps in sleep regulation. More melatonin, good sound sleep you get into. Okay. It's secreted from the pineal gland. Melanin is a pigment. It's a protein which is secreted in our epidermal cells, which gives us color, the skin, the color. It's not present inside the body. Our inside organs are not, they do not have color. Okay. Like they may have their distinct coloration and they may have their pigments, but not the skin kind of color. So write down. So basically melanin is a protein, yes. And I told you that DNA, genes are present in the form of DNA, right? And DNA makes RNA, right? And RNA makes proteins. Yes or no? You have discussed this. So there are ribosomes in the cell which read the RNA and make proteins. They are called protein factories in the cell. So if melanin is a protein, for melanin also there must be a gene, right? Now,
Yes.
if there are more than one genes, more the number of genes, more is the number of RNA, and more will be the protein. Yes.
Yes, sir.
So darker will be the skin shade. You understand?
Yes.
Apart from just genetics, there is something else which affects our skin color, and that is environment. You know, direct exposure to the sunlight. Yes or no? We get tanned when we are in sun, even when our genetic composition is same. We cannot, it's not like we go in the sun and our genes change, right? That is not happening. Yes or no?
Yes, sir.
But our skin color does change. Or if someone, so you will see that a couple who is based in Africa, they are an Afro couple. So probably they will have more melanin in their body, and they will have multiple genes which are active for melanin production in their body. Yes.
Yes.
So their baby, when it will be born, it will be having a darker skin shade. Yes or no?
Yes, sir.
And no matter what, if you send that, so if it has a darker skin shade, already lots of melanin is there. In just one generation, you cannot lose all that genes. But if you send that baby right from birth to live in Europe, so over time, over generation, that person's generation, even if that person continues to marry a African woman who also established there, but over time you will see that there will be slightly lesser skin, fairer skin shade coming in that family. Yes or no? Because now they do not need that melanin. So there will be kids who will be little fair, one shade fairer than the parents. Yes or no?
Yes, sir.
And on the other hand, if you have someone's kid, like say in Europe, and when the kid is born, it's very fair. But if you send that kid to live in Africa or in near equator, any country near equator, since childhood, then as it will grow, the skin tone will become darker and darker, right? More and more melanin will start producing. Yes or no?
Yes.
To protect itself from the sunlight, correct? And the generations coming.
Yes.
So write down what is polygenic inheritance. Polygenic inheritance. Polygenic inheritance is where traits are controlled by 3 or more genes. Traits, any trait, skin color, height, etc. Traits are controlled by 3 or more genes and also are influenced by the environmental factor and also are influenced by the environmental factors. You understand, everyone? Do you understand?
Yes, sir.
People. Done? Dictation is done. So write down some examples are human height and human skin color, or height or skin color in humans. These are polygenic traits. Human height and skin color in humans are polygenic traits. Human height, human skin color. Okay. Now let's assume, to understand it better, let's assume for human skin color, right? Let's assume there are 4 genes responsible. Okay.
Yes.
And the 4 genes are gene A, B, C, and D. So if all the genes are like expressing, then there will be more and more melanin. Okay. If all 4 genes are active, which means if the genotype is this, capital A, B, B, C, C, and D, D. So in this will be the darkest skin. You understand why? Because all are expressing and more skin pigment is being produced. And which will be the fairest? Where all the genes will be recessive. Yes. This genotype will be the fairest skin. Now they can also be influenced by environment, but we are not taking environment into account now. In between, you will have many combinations, right? For example, AA, BB, CC, but small d. You know, there is one gene which is recessive now, homozygous recessive. Or if there are 2 genes which are homozygous recessive, you understand? Then this will go like this. You understand?
Yes, sir.
So there are many shades possible. As many permutation combinations are possible, as many shades are possible. Make sense to all of you why humans' skin's colors are very, very different and comes in all shades?
Yes.
Yeah. So more number of genes dominant and homozygous, more is the pigment and more darker is the skin. Similarly, more exposed to sunlight, more darker is the skin. Yes.
Yes.
So environmental factor also plays an important role. Polygenic inheritance is clear? Any doubt?
Yes.
I'll take the last topic for today is just opposite of polygenic inheritance, and it's called pleiotropic inheritance or pleiotropy. Now, in polygenic inheritance, we saw that there are multiple genes for one character. Okay. In pleiotropy, it's opposite. There is only one gene, but it affects multiple characters. Again, not according to the common laws of Mendel, right?
Yes.
Yes. So write down pleiotropy. Pleiotropy is where a single gene. Pleiotropy is where a single gene can exhibit or influence multiple characters or multiple phenotypic expressions. Phenotypic expression simply means characters. Okay, everyone?
So
So such a gene will be called a pleiotropic gene. Pleiotropic gene. It will be called a pleiotropic gene. And one such gene example we see. Let's go to an example. Just like in this, the example was human height and human skin color. Here the example is a diseased condition which is known as phenylketonuria. Phenylketonuria. It is a diseased condition which occurs in human. Okay, so a disease that occurs in human. Okay, Now, basically, this disease happens because of a mutation in a single gene. Is it clear, everyone?
Yes.
So if there is a mutation in a single gene in body, and this is where genetic becomes more complex. Remember, did we discuss that there was a Chinese scientist who edited human genome and removed a gene for obesity, which causes obesity, but he was jailed instead? Did we discuss that?
I think we discussed it.
Yeah. And why he was jailed? Because I told you that, so someone said that he did a good thing, right? He made a human who cannot get obese now. So there will be no disease, obesity will not be there in the kid. So what's the problem? The problem is that we do not know whether that one gene is just having one effect, which is just causing obesity, or it is having some other effects on other things as well. Because if that gene is pleiotropic, then it may also be playing role in other things, right? And maybe some of that is crucial. Yes or no? And anyways, doing experiments on humans is illegal. You understand? So in phenylketonuria also, there is a mutation in a single gene. So if there is a mutation in a single gene, and this gene, write down what does this gene do. So this gene codes for an enzyme. And that the name of the enzyme is phenylalanine hydroxylase. Yes, phenylalanine hydroxylase. So this is the enzyme which gets mutated. Okay. So gene gets mutated. So if there is only one gene getting mutated, you will see that, you will think that, okay, this is one gene. So maybe one trait will get affected, right? Yes or no? But that's not true.
Yes.
This gene, which codes for phenylalanine hydroxylase, plays important roles in multiple characters. That's why when phenylketonuria happens, write down the phenotypic expressions of the disease. The phenotypic expressions of the disease are. So one gene is faulty. For example, do you know about diabetes, people? Diabetes as a disease, Atif, Fatima?
Yes.
What is diabetes? What happens in diabetes? Let's say there is a gene. A diabetes has happened because of a gene defect. So that gene will be insulin gene, right? Insulin is not being produced properly, or that gene is faulty. So faulty insulin is being produced, which is not working in the body. So person get diabetic. So that kind of a person will be diabetic right from the birth. Yes. But there is one gene, insulin, and it is causing diabetes, and blood glucose level keeps rising. See that. But in this disease, phenotypic expressions of the disease are very, very vast. First thing that happens in this disease is mental retardation. Now you will think that, okay, mental retardation means brain is not forming properly, correct? Or it's not functioning properly. The connections in the brain or the cycles in the brain are not functioning properly, right? Mental retardation has happened. But in this disease, you will also see reduction in hair pigment. Now, do you think the gene for hair pigment and mental well-being should not be the same one? Yes. And also there is reduction in skin pigmentation. So don't you think these are very, very different characteristics which are getting affected by one single mutation in one single gene?
Yes, sir.
Yes. So this proves that this one single gene which codes for this enzyme, phenylalanine hydroxylase, actually is a pleiotropic gene. It plays a role, or it affects, influences a lot of different characters: mental well-being, formation of neurons properly, growth of brain, pigmentation of the hair and pigmentation of the skin in humans. So it does all of that. Do you understand? That's why when that got faulty, all these things became problematic. Okay, is it clear? Any doubt, people?
No, sir, no.
No doubt. Okay, so we will do such disorders which are like Mendelian disorders, but these are just two topics, pleiotropy and polygenic inheritance, that you should know. So this is where I end the class today, and I'm giving you a homework for the before you come in the next class. You have to go and read about. We already know about sex determination in humans, right?
Yes.
But you have to go and read about sex determination in human, chicken, an insect. Let's take an example of a cockroach, and let's also take birds, any bird, and a reptile. So there are many different types of sex determinations in nature. Many of them are genetic, many of them are genetics plus environment. So that we will do the next in this chapter. Then we will do, after sex determination, we will go for Mendelian disorders, chromosomal disorders. Okay.